Good morning, children. Today's topic is microsporogenesis. See, these are the four microsporangia in which sporogenous tissues are present, and each cell of this is diploid in condition 2n. And these are known as pollen mother cells, PMC, or microspore mother cell. Now pick up one PMC, pollen mother cell, which is diploid in condition 2n. One meiosis will take place. Only one meiosis division will take place, but we know that in one meiosis two phases are there, meiosis 1 and meiosis 2. So meiosis 1 is reductional division, so two cells will be formed, but they will be haploid in nature. From 2n it will become n. Now meiosis 2 will complete and four cells will be formed, which will be known as microspore tetrad. The process of formation of the microspore tetrad from microspore mother cell is known as microsporogenesis. Now let us study the structure of microspore or the pollen grain. Now see, this is the tetrad. Pick up one to understand. So this is the nucleus and this is the plasma membrane. One mitosis will take place. As mitosis take place, so from n it will remain as n. Both two cells will be formed, one known as vegetative cell, which will be bigger, and the other one, which is spindle shaped, will be known as generative cell, which is small. So this is the two celled stage. Let us study the structure of this microspore. Outer to plasma membrane, intine will develop, which is made up of cellulose and pectin. Outer to that, exine will be formed, which is made up of sporopollenin, which is the most resistant organic chemical. No acid, alkali, enzyme can act on it. Even the high temperature will not break it. So this is the most resistant organic chemical. Therefore, the fossils of pollen grain can be formed. In most of the cases, these exine, they are beautifully sculptured. Okay, this is not continuous. There are germ pores present. In most of the angiosperms, 60% of the angiosperms, the pollen grain, the microspore, is shed in this two-celled condition. And once they are shed, they will land on the stigma of the flower, and from this intine, from the germ pore, the intine, the pollen tube will develop. And now one mitosis will take place in the generative cell, and two male gametes will be formed. But they will be shed in two-cell condition. This will happen when the pollen grain will land on the stigma of the flower. In 40% of the angiosperm, the pollen grain is shed in three-celled condition. That means before they are shed, in the generative cell, mitosis will happen and two male gametes will be formed. So now this is in three-celled condition.